All right. Last lecture. We made it. You made it. Good job, guys. Be happy. Be impressed with yourselves. All right. This one's fun. It's going to be on the shorter side, a little bit more direct, like the last couple. Uh, this one, though, it has some pretty important consequences. So unfortunately, you'll have to see me get on a soapbox for this one. It's one of the few soapboxes I get on while teaching this in nuclear energy. So um, it'll just be kind of like stewardship conversation at the end. All right, so let's get started. Lots of key terms that look like drugs that you've heard before, hopefully, at least some of them. But let's dive in. So, antibiotics are honestly miracle drugs. Prior to antibiotics, you could get a cut and die. And now we don't die from simple cuts. You could get an infection as a young child and die. So antibiotics, if you look at a graph of the population of the world of humans, right around the 1940s, the population goes from pretty static to suddenly an explosion in growth. And it literally looks like exponential growth. And that's partially because of antibiotics, partially because we also at the same time learned sanitation is important, how to clean our food and water better, how to do surgery without killing the patient, but mainly antibiotics extended lifespan. So people were able to survive into adulthood and have children of their own. But we're facing a really big issue with antibiotics right now where they're becoming less and less effective. We'll talk more about this later, but that drug resistance is a huge problem. One thing that people don't think of with antibiotics is ancient times. People think antibiotics are a fairly new thing, but antibiotics have literally been around for billions of years because they are made by microbes to attack each other. They're kind of like the weaponry that they use in the fight for, resources and space. It's their way of out-competing each other. So antibiotics are just compounds made by a microbe that can kill or inhibit the growth of other microbes. That's the technical definition of an antibiotic. But we also use this to discuss the artificial or synthesized antibiotics that we've created. Because technically, we've never created brand new antibiotics that we've never seen before. What we tend to do is say, you guys made something really cool. We're going to modify it a little bit and call it a new antibiotic. So, with this, it's really, when we talk about it, we're going to talk about every single type of antibiotic, natural or clinically synthesized. But with humans, even, antibiotics have been used for thousands of years. We just didn't really know what we were doing. We kind of said, well, this is working, so we're going to keep doing it, but we don't know why it's working. So there's lots of written evidence of ancient China, ancient Greece, ancient Rome, ancient Egypt. They used to take soil or they used to take bread mold and they'd pack wounds with it, and people would not get infections as a result. They didn't know that the mold or bacteria in the soil were producing antibiotics that would stop any kind of infection in your wound. And then on top of it, technically Nubian, but basically Egyptian bodies have been found where there's tons of tetracycline in their bones, almost like they eat it as a natural part of their diet for their whole lives. And what we found was probably they were using a special fermented beer that they would drink at nearly every meal. And it had Streptomyces bacteria in it. And when they ferment, they produce a lot of tetracycline. So there's some evidence that either this was being consumed all the time, and that's why it built up so much in their bones, or it would be given as a treatment in high doses to people. So we've been using antibiotics for quite a long time, but the modern thoughts of antibiotics began in the 30s, really. So we talked about Alexander Fleming a little bit. He discovered penicillin in 1928. And then it was later in the 30s purified and given to people for medication. But penicillin is only effective against gram-positive bacteria. Gram-negative bacteria have too many defenses against penicillin and those types of antibiotics, the beta-lactams. So it's only effective against some of the bacteria you can get sick with. Then in the 1930s, Gerhard Domach, he discovered sulfa drugs like Bactrim. These are very commonly used. And then Semon Waxman in the 40s discovered the streptomycin bacterium. These are broad spectrum. So not only could they kill gram positives, but they can also start killing gram negatives. So now we had coverage against all types of bacterial diseases. But even with this, antibiotic resistance already started appearing as early as the 1940s. Technically, it probably was appearing from day one. We just didn't notice it yet because, once again, these antibiotics are made by other microbes. So microbes for billions of years have already had to counteract these antibiotics. So resistance is not just a problem that we created. It's a problem we are making worse, but resistance already naturally occurs in bacteria all the time. And we'll talk about what some of those mechanisms are at the end. This led to what we call the golden age of antibiotics, where basically all through the 40s, 50s, 60s, 70s, we were discovering or making new antibiotics. And that has significantly slowed down. We're having a much harder time finding antibiotic compounds now. So Dr. Ruiz's lab that I worked in, one of the goals that he has is to kind of get around this idea of antibiotics and say, okay, well, bacteria use these efflux pumps. If we can find some other chemical that's not an antibiotic that can block the efflux pumps, it'll make the bacteria more susceptible to the antibiotics we already have. So there's lots of research out there that's saying, okay, we're having this antibiotic resistance problem. How can we get around it to make sure that our antibiotics still work well? So that's a really big area of research. If you want to go into a lab or start a lab, that is always going to have funding. Antibiotic resistance research is definitely that. Dr. Ruiz just got a $1 million grant from the NIH when no one else here can get a grant. So it's a very important, very, very important problem, and people are throwing money at it like crazy. All right, so antibiotics work because they exhibit selective toxicity. So they are toxic to the microbe that you want to kill, and usually, hopefully, ideally, not toxic to you. Bless you. So they're not going to hurt us or as much as possible our gut microbiome or our skin microbiome. We just want to inhibit the pathogen as much as possible. So antibiotics tend to target very specific things that are in microbes that are not in us. Things like peptidoglycan synthesis. These are only in bacteria. So we don't even use antibiotics that affect peptidoglycan against protists or parasitic infections because they don't have peptidoglycan. They won't work against it. So this is a very specific bacterial target. We can also target the difference in ribosomes. So remember, prokaryotic ribosomes are 70S, whereas eukaryotics are 80S. So if we can target the differences, something that only affects 70S and not 80S ribosomes, you're good. However, these drugs tend to have a lot of off-target toxicity, meaning they do cause us to get sick too, even though we have an 80S ribosome in our cells. Why do you think these might have off-target toxicity? You can talk to your neighbor about it, but partially, this is actually saying it hurts our cells directly, not just our microbiome. So all of these are in one way or another going to harm our microbiome to an extent. So when you take antibiotics, you usually have to make sure you're doing things to support your microbiome as well. But this one's saying it actually hurts our cells directly. Good guess. Not quite, though. No, that's a good thought. But think about something that lives inside of our cells. Our mitochondria, they have 70S ribosomes. They are prokaryotes living inside of us. So these ones will actually affect our mitochondrial health and cause toxicity to us as a result because they're effectively part of us. So we need them to have proper energy and metabolism and things like that. And then we can also target biochemical pathways that bacteria do that we don't. There's a lot of those. One example is folate biosynthesis. So humans cannot make folate on their own. It's a big reason why pregnant women have to take it as a supplement because babies consume a lot of it when they're gestating, and we can't make it on our own. You have to get it all from eating, or you have to get it from the bacteria in your gut. A lot of our gut bacteria will make folate for us. They will use some of it and we will use the rest of it. So if we target a biochemical pathway that only bacteria can do, that they need to survive, then they're going to be killed, but we will be fine. We might have a little bit of a nutrient deficiency for a little while, but as soon as it's done, we'll just eat some more folate. All right. Now, antibiotics also have what we call a spectrum of activity. There's broad spectrum drugs. These ones can harm many different types of bacteria, different species, gram positive, gram negative. Then there's narrow spectrum. These can sometimes harm only one species of bacteria, sometimes maybe just one little group or genus of bacteria. So it just depends on the drug itself. Vancomycin is an example of this. It really only hurts gram positive bacteria. Isoniazid, that really only interferes with mycobacteria like tuberculosis and things like that because it interferes with their mycolic acid production. If you're a gram positive or a gram negative, you don't make mycolic acid, so that's not going to have any effect on you. And then we classify them one other way. Not only can they be broad or narrow spectrum, but are they bactericidal or bacteriostatic? So bactericidal, that cidal word, we tend to put it with homicidal, you're killing someone. So these are the ones that are going to kill the cell that you are, the pathogen. So bactericidal antibiotics kill the cell directly, whereas bacteriostatic ones, these just stop the growth. Why might we want to stop the growth? How does that help us clear an infection if we're just stopping growth rather than killing them? They can't keep reproducing, but they're still in us and they can still cause disease. So what else does it do for us? That's half of it, yeah. Yeah. If they're not growing, they can't overcome our defenses. If they're not increasing in population, then we have time to mobilize our defenses and thoroughly kill them. So if we limit their growth, we can take them out easier. It's kind of like if we take out all the boats of a country that lives across the ocean or all the planes of another country, they can't get their troops here to hurt us. So whatever troops we have here are easier to take out. That's the idea. That's a good question. I have never heard that argument for them because we also have bacteriostatic that affect gram positives. But yeah, that could definitely be part of it. If we're limiting their growth, then there's less lipid A, less intense shock if when we do start killing them off. That's a good point. But I've never heard that argument directly. Um, there are many different ways that antibiotics work, and we're going to go through a bunch of these examples. We can target the synthesis of the cell wall because there's peptidoglycan there. The cell wall is a crucial component for the cell to live. If we break the cell wall, they can't survive that osmotic pressure, they burst and they die. So this is a really good one to be able to tackle. We can also target their cell membrane. The problem with this is their membrane is identical to our membrane. So if we do target their cell membrane, we're probably going to harm ourselves too. These are kind of our last resort drugs, where if everything else fails, it's kind of like, I don't care if I hurt myself too. I just want to try to kill this thing whatever way possible, like the SOS mechanism of repair. There are some ways that we can target the cell membrane that don't harm our cells quite as much, but they will harm our mitochondria pretty badly too. So we can use things like detergents that directly degrade the membrane, or we can make them leaky to ions so the cell loses its proton motive force capabilities, things like that. Then we can target DNA synthesis. Their polymerases, their topoisomerases are very slightly different to ours, just a little bit. So sometimes these have off-target effects as well because they are similar enough, but the differences allow it to be a target as well. Same thing with RNA synthesis. We can target the 70S ribosome and target that protein synthesis, or we can target parts of metabolism that we don't do. Any of these can either slow the growth or stop the cell from living. So something like protein synthesis, that's not going to directly immediately kill the cell. It's just saying you can't make any more protein, so you have to live off of what you've already got, which that's fine. The cell's eventually going to die, but in the meantime, it's still alive. Whereas something like cell wall synthesis, if we break apart their cell wall, they're going to lyse pretty quickly. That's going to directly kill them. Any questions about that? It's cool stuff to finally learn how all this works, right? We take antibiotics our whole life. So here's just some examples of ones that fall into all those categories. You've probably heard of or taken some of these, and we're going to go into a few of them now. All right, any questions about that? With these slides, these next two slides, I am not going to test you on any of this. This is more reminder slides because we're going to talk about antibiotics and what they target in these processes. So I want to just go over a couple key things regarding cell wall synthesis, but you will not have to explain any of this to me. So we have peptidoglycan made from NAG and NAM and then the peptide that connects them. You do have to know that for the exam, but that's because of the old stuff. However, these things have to be made too. So they have special kind of like carrier molecules or stabilizing molecules that help them get built up. They are going to be attached to the precursors to a lipid called bactoprenol. And the bactoprenol, once it's made, takes this NAG and NAM and flips it to the other side of the membrane where it needs to go. So inside the periplasm or the external environment. So we've got these precursors, they're made, and then bactoprenol puts them where they need to be. And then we continue to take those precursors and stick them together to make the whole peptidoglycan. Part of that is the transglycosylases, those link NAG and NAM together. And then we have the transpeptidases that form the cross links between the little peptides. We're really going back, like day one of class almost. So bactoprenol can be a target, the transglycosylases can be a target, and the transpeptidases can be a target, because each of those is a crucial step towards synthesizing the cell wall that if we block it, it can't make the cell wall. It either can't put it where it needs to be, or it can't connect the pieces together, so it's unstable. That's the whole idea that I want you to get from this slide here. You won't have to explain any of that to me. Don't stress. Okay. We're going to talk about these a little bit more later, but now that we just went over the cell wall, let's discuss some of these. So there's cycloserine. This inhibits the assembly of the peptide chain. So we add the peptide, the amino acids one at a time, and this will block the ability to add more of them. Then we have bacitracin. This is a topical antibiotic, and it inhibits the recycling of bactoprenol, the one that helps us flip it to the other side. Bactoprenol has to then come back and regather before it can work again. So if we prevent it from coming back and being recycled, the process of flipping them to the correct side is halted. This is made by species of Bacillus bacteria, and it was first found in a skin infection on a woman named Tracy. So that's why it's bacitracin. I just kind of thought that was silly, but we do not ingest this one because it's only able to work topically. And then we have the beta-lactams. We're going to get into these guys a lot. These are your penicillins, basically. They inhibit the transpeptidases, so they prevent the cross-links from forming. We talked about that way back in the day, too. But we're going to get into a little bit more. So those are things like ampicillin, penicillin, amoxicillin, cephalosporin, vancomycin. I bet you every single person in here has taken one of these at some point in their life, unless you're allergic, and then you found out because you took it and you had an allergic reaction. Amoxicillin is a big one for like strep throat. All right, any questions before? Yeah. It's basically that our immune system sees these compounds and is like, foreign invader, and they start attacking it and causes you to have a massive immune response to it, and you die from the immune response. So it's the same way that you can develop allergies. Instead of attacking a foreign invader, your body starts attacking the wrong thing. And it's not supposed to attack these compounds, but it decides, nope, this is bad, we have to attack it now. And that's completely random who that can happen to. We haven't really found any genetic predispositions to allergies to different drugs like that. It's more just your immune system decided it would get a wild hair up its butt one day. Just like with autoimmune diseases, which really suck. So let's get into the beta-lactams a little bit more. So this is basically the penicillin class of antibiotics. And their main mechanism of action is that they all have a beta-lactam ring. And you can see it right here. It's a little square. So it's a 4-carbon ring structure. And what we do is we take this basic structure of penicillin and we will modify around the beta-lactam ring. And that's how we create new generations of beta-lactam antibiotics. So we can add functional groups to them and create a form that the bacteria aren't resistant to. The problem is you can only modify something so much and have it still function properly. So eventually bacteria can evolve resistance to those new forms too. So the way this works basically, did I say it on the next one? No. This ring structure here, it can basically interfere with the transpeptidases. It can bind to the active site of the transpeptidases and prevent them from being able to function. And it binds to them pretty tightly. So once they bind, they don't usually come undone in any hurry. So if we're a bacteria and we're being attacked by a beta-lactam, and the key portion is that ring, one of the main things I want to be able to do is have an enzyme that can break that ring somehow. So that's one way that we can counter, as bacteria, they counter these beta-lactam antibiotics. All right, cool. Now we can, as I said, modify these, and we've created things called cephalosporins, which are kind of like the lab-made penicillins. And here's just a bunch of different versions of these. The first generation was like Keflex. I don't know if any of you have taken that. It's pretty common to be given to kids who have like face issues of like acne or skin conditions on their face or anything like that. I had this as a kid, and I think I'm allergic to it. I don't know. I took it for an infection, and then I broke out really badly, and the doctors are like, that's like a mild allergy. I'm like, okay. So I can't take that one. But Yeah, if you notice, they start out somewhat simple, and then we kind of increase the complexity with further and further generations, because we have to keep tricking the bacteria and saying, no, 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 you haven't seen this before. It's not a beta lactam. Don't worry. But yeah, we just increase the complexity over time. Now, vancomycin is in the same family, but it works a little bit differently. Instead of binding to the enzyme itself, it will bind to the peptide side chains. So the transpeptidases will take the peptides of NAG that are attached to NAG and NAM and connect them. And so if the enzyme can't get there because either the beta lactam is inhibiting the enzyme directly or vancomycin is attached to the peptide, then we can't form those cross links, and the cell wall stays pretty loose and can be targeted for killing by oxidative stress, or not oxidative stress, osmotic stress. So it's almost the same as the penicillins, except instead of binding to the enzyme, it binds to the peptide. And then we talked about cycloserine a second ago. This inhibits the D-ALA formation. So when we're trying to make that cross-link, we have two D-ALAs and we cleave one of them. So in the beginning, we have to put the two D-ALAs together. This one prevents that from happening. So it's kind of inhibiting the step before cross-linking. And then bacitracin is going to keep those precursors from ever getting to the periplasm, so we can't even start making the peptidoglycan at all. Any questions about those? I talk about these because they are some of our most common antibiotics, the ones that have the least toxicity to humans, so we tend to give them out a lot. And they're very helpful, but we're getting significantly more and more resistance to these guys, and it's getting bad. Okay, let's talk about different types of antibiotics now. So we're moving a little bit away from the cell wall here, and we're going to talk about the membrane just for a moment. So the first one, and the only one we're going to talk about, is gramicidin. This guy is a peptide made by Bacillus brevis. And what this does is this whole little peptide forms a pore. And that pore gets placed into the membrane. And it's basically a leaky channel. And all the ions start moving freely through this channel. And it basically destroys the concentration gradients that the cell is working so hard to make. So it puts everything at equilibrium. Now there's no proton motive force, so the cell can't bring in nutrients, it can't make ATP, it can't move its flagella, and the cell is really hurting at this point. It probably dies pretty quickly. So we're basically killing it by poking holes in the membrane. Any questions? Would you expect this one to harm us as well? Would this have off-target effects, basically? It could. Yeah, it could, because this is targeting the cell membrane. And even though, you know, our cells are bigger and we've got a lot more stuff to protect ourselves with, we're still susceptible to leaky ion channels too. Even more so, our mitochondria are. So this one we want to try to not use very frequently because of those off-target effects. And a lot of times, ones that target the membrane, we will only use topically, because it's really hard for those to actually penetrate our skin because of the keratin layer. So it's going to just be killing the bacteria right in the area that we need it targeted, rather than all throughout your body. All right. We also have polymyxin and daptomycin. Polymyxin is a polypeptide, so it's protein-based, made by Bacillus polymyxa. That's where it gets its name. And it acts very similar to a detergent, and it completely disrupts the cell membrane and breaks it apart. And we only use this topically, usually in a form called polymyxin B, because these are extremely toxic to people. These are kind of like last resort drugs because they are so toxic to us. One in particular called colistin. This is one of our last resort drugs for gram-negative infections that are resistant to lots of different antibiotics. So in particular, one of them is by Klebsiella pneumoniae. So Klebsiella pneumoniae is a relatively recent massive threat to humans. About a decade and a half ago, there were a bunch of cases of resistant Klebsiella pneumoniae, and so we started to treat it with colistin, this particular form. And then maybe like 4 or 5 years ago, we had a bunch of people die because Klebsiella pneumoniae is now becoming resistant to even colistin. So kind of like the cancer saying F you to the chemo. It's like, this was our last resort. It's already killing the patient too. And now that doesn't even work. And that's kind of scary because Klebsiella pneumoniae is kind of found everywhere. Granted, the people who are dying tend to be children or the elderly. So it doesn't really harm people in the prime of their life who have strong immune systems, but we are all going to be elderly one day, and many of us will have children one day or have children. So that's really the target audience for that type of infection. And it's kind of scary that we can't treat it, because then eventually it could become even more pathogenic and start harming healthy people too. It also is used on Pseudomonas and Acinetobacter because they're pretty, in general, antibiotic resistant species. And then we've got daptomycin. This is a lipopeptide, so it's a protein with a little fat on it. And this is made by Streptomyces roseosporus. And what it does is it forms an ion channel that causes potassium ions to leak. And we tend to use this one a lot against MRSA, methicillin-resistant Staph aureus. Skin infection. Me too. It's fun, yeah. Yeah, you can be carriers of lots of nasty things. My dad was a carrier of strep, and so he would give it to me a lot as a kid, and I would get strep infections like 6 times a year because he always had it. And if you live close to people, you're going to get it from them. Nope, he was just kind of used to it. It was kind of at this point part of his natural microbiome. Sucked for me, but all right. So before we move too much further with this, I want to make clear what to expect for the exam regarding this stuff. I'm not going to ask you what is the chemical structure, so you don't have to tell me it's a polypeptide or a lipopeptide. I'm not going to ask what bacteria it's produced by or what fungus it's produced by, anything like that. I will ask how they work. So you'll have to know that a polymyxin destroys the cell membrane. You'll have to know that the beta-lactams, that ring, inhibits the transpeptidases. So you got to know mechanism of action for all these antibiotics, not all the other details about them. That's just more to round out the story and help you if you decide to go into a clinical setting.
So this is for all, like,
all the ones that we're going to talk about. Yep. All right. Let's talk about ones that, yeah, sorry. Yeah. No, this one we don't use topically. Yeah. So colistin is not, but polymyxin B is only used topically. Yep, good point. Thanks for clarifying that. All right, so drugs that affect DNA synthesis in bacteria. We have several classes, but 2 that you've probably heard of or know of are the quinolones or the fluoroquinolones and metronidazole. If you work in a vet hospital, I know you've heard of metronidazole. We give that out like candy. So the quinolones, though, these are compounds that naturally occur in plants. And what they're going to do is block bacterial topoisomerases. So it blocks the gyrase that helps relieve that twisting stress on the DNA. And as a result, it disrupts DNA replication. If we can't relieve that torsional strain, the DNA polymerase can't move forward in the replication fork. It can also induce double-stranded breaks in the DNA because of the strain. So this is pretty dangerous to the bacterial cell. They do not like it. One example of this is nalidixic acid, but more commonly we use the next generation, the synthetic versions like ciprofloxacin. How many of you have taken one of those? Yeah, they're pretty common. And then you've got metronidazole. This is what we call a prodrug. So the metronidazole itself is not able to kill anything. It's not able to harm anything. It has to be processed in your liver before the active form is released. So our body physically breaks down this compound into the active form where it can then get into our bloodstream and do its work. So it forms what's called a nitrous free radical. So instead of an oxygen free radical like superoxide, this is the next worst kind, not the absolute worst, the next worst. So a nitrous free radical. And what this will do is basically oxidize a bunch of your proteins that are involved in DNA replication, and that's not good. It also, this nitrous free radical is used by the cofactors for ferredoxin and flavodoxin, which are important for bacterial metabolism and iron acquisition. So if they can't do DNA replication, if they can't get iron, if they can't do metabolism, how are they going to live? We tend to use this most against anaerobic bacteria because aerobic bacteria often have ways of counteracting this, because they're already used to all these free radicals. So they'll have the enzymes like catalase and things like that that can detoxify it. So we use it against anaerobic bacteria, which are often gut bacteria, and protozoas. So those are often ones that are going to infect the gut. And so you often see this given for diarrheal problems. Yeah, you know. Yep. It's like, oh, my puppy is vomiting a little bit, metronidazole. My puppy, his stool was a little loose, metronidazole. Yep. It works well, though. And because dogs tend to get a lot of protozoa, they'll drink water from all over the ground that has like giardia in it and nasty stuff like that. So we don't tend to get a lot of protozoal infections as humans because we drink treated water. But animals who don't drink treated water, they tend to get more of those. All right. Then we have drugs that inhibit RNA synthesis. So now we're looking at inhibiting the RNA polymerase somehow. So with these ones, they are bactericidal, so they are going to kill the bacteria. And these often work best on cells that are currently in like exponential phase, because we're targeting cells that really need to copy DNA into RNA, do transcription, and usually that happens the most in exponential phase when they're changing up their expression, when they're making the proteins that they need to make new cells. We tend to lower the amount of transcription that we do when we're in stationary phase. If you're a bacteria, I keep saying we are not bacteria. Sorry. One of these is rifampin. So this is going to bind to the RNA polymerase itself, and it prevents the elongation step. Now, even though our polymerases are very conserved, bacteria know that this is a target, and it's a very important target. So they can actually allow this to mutate very readily. So resistance to rifampin occurs very, very quickly. Bacteria will allow mutations to run rampant so that they don't have this being blocked. So usually we either can't give this antibiotic alone because resistance develops to it so quickly it's not enough, or we can't give it for long periods of time because it'll encourage resistance to develop. So who we often use this for is Mycobacterium tuberculosis. All of the mycobacteria are incredibly slow growers. I mean, like, if E. coli is doubling every 8 to 15 minutes, these are doubling like every few weeks. So they're very, very slow growers. So this gives them time to be killed, but they can't mutate fast enough for it to become a problem. And then we have actinomycin D. This does not directly block the polymerase. It's going to block the DNA, so it blocks the polymerase from binding to the DNA, basically. So it binds to DNA anywhere. It doesn't have a specific spot that it likes to bind to, but sometimes it happens to block an important gene. The polymerase can't bind, it can't transcribe, and you can't make that protein. So it's a planar molecule, so it's very flat, just like we talked about with ethidium bromide. So it'll fit in between our DNA, and it'll actually, it can induce double-stranded DNA breaks as well, but it just mainly prevents the polymerase from being able to bind. So this part will fit in between, and these will stick out of the DNA, and polymerase is like, I can't reach the DNA because these guys are in the way. This is going to prevent initiation because it's blocking the RNA pol. So this prevents elongation. This one prevents initiation. I'm waiting because I see people writing still, and we've got time to wait today. So sorry for all the pauses today. All right. Then we've got the protein synthesis inhibitors. These are the ones that are going, oh, question? Oh, I thought you. These are the ones that are going to block the 70S ribosome. But these ones can affect our mitochondria as well, so we have to be careful with those. Usually they're going to bind to and block the ribosomal RNA. So that's the target there. And usually these ones are bacteriostatic, not bactericidal, because once again, you can survive for a time without making proteins. You can't really survive very long if you can't make DNA. You can't survive very long if your cell wall is broken. But you can kind of make do. You just won't be able to grow if you can't make new proteins. So, two things we can target, the 30S or the 50S subunit. So the 30S subunit, these are your aminoglycosides and your tetracycline antibiotics, and these are very, very lethal to the cell. So what they do is they create something called slippage, meaning as the ribosome is trying to read the mRNA, it keeps like sliding on the mRNA, and it can't get a good grasp on the mRNA. So as a result, that causes the ribosome to make many small peptides. Small peptides are surprisingly very, very, very toxic to bacterial cells because a lot of them act in a very hydrophobic manner. So they'll be drawn to the membrane, they'll insert themselves in the membrane and basically disrupt any type of concentration gradient at the membrane, disrupt the membrane itself, and the whole cell integrity falls apart. So this actually kills the cell very quickly as a result. So if you've ever taken Neosporin and put that, that's a aminoglycoside. So that's a topical version, but you also have streptomycin, gentamicin, amikacin, and then tetracycline and doxycycline or others. These two you tend to give for Borrelia burgdorferi infections, so the ones that come from the ticks that cause Lyme disease. And resistance to this has not really been a problem. The bigger issue with this is production. We can't produce enough of it often. We've had a lot of big, what's the word I'm looking for? We've had issues where there hasn't been enough for demand recently. With the rise in Lyme disease, it was being given a lot for secondary infections with COVID. So we had a lot of big supply chain problems with those during COVID as well. And then you have some that target the 50S subunit. These ones are bacteriostatic. They're not lethal like it is for the 30S ribosome. These ones are just going to interfere with that peptidyl transferase activity, so we can't grow the polypeptide. We can't continue making our protein. So these are your macrolides like erythromycin or azithromycin, if you've ever taken a Z-Pak, which I'm pretty sure nearly all of us in here have at some point. That's a macrolide. You have the lincosamides like clindamycin. That's another one that we give out to kids and puppies. It's often in a liquid form. And then we have chloramphenicol. This one is not really used in the US anymore because it can cross the blood, or not because, it has mitochondrial toxicity, sorry. So this one it's dangerous for us to use because it harms our mitochondria, but it's a really great drug. It's so stable temperature wise. You don't have to store it any specific way. You can take it into really hot, wet, humid jungles and it doesn't break down and it stays stable for long periods of time. And it is cheap. So it's used all over the world, just not really with us. It also is one of the few drugs that can cross the blood-brain barrier, so it can also cross into the placenta easily too, and we can use it to treat encephalitis, particularly in young kids. But for some reason, we just don't like to use it here because of the toxicity. Our attitude is, we could use another drug first. But, oh well. And then things like linezolid, those are also bacteriostatic ones that target the ribosome. That's much less likely to be given. I don't think I've ever heard of someone being given that one.
Like, is that it's not being used, but there's still a stock of?
Oh, yeah. That's the other issue with my antibiotic resistant soapbox. We used to make a lot of antibiotics here. Now China makes nearly all the antibiotics for the whole entire world. So if we get into a war that drags China into the war with us, we could potentially experience massive antibiotic shortages here. So why China? Because they have fewer regulations on human rights, so they can manufacture things for cheaper and faster. Yeah. Especially where we're using this a lot. It's not necessarily like, outside of the US, so this is being used in Germany and Italy. No, it's used in areas that are very, very poor, where there's not a lot of medical adherence anyway. So at this point, if the kid's coming to you, the encephalitis is really bad. So it's like the SOS, just treat it no matter at what cost. Yeah.
Mm-hmm. For example, so we're just talking about like 40-30 ads that you did, put it effect, and then
yes, exactly. And you do have to know that an aminoglycoside is one that'll target the 30S. The tetracyclines target the 30S. And that how they work is by making it so the mRNA can't be read, and that disrupts the membrane. Mm-hmm. Now, how do bacteria combat all of this? Because they need to have ways of fighting this if they're going to survive. Everything wants to survive. It doesn't want to die. So, number one, we can keep the antibiotic from getting into the cell. We can have mechanisms to block it from ever entering and causing havoc. Two, they can prevent the antibiotics from binding to the target. We can modify the target so suddenly the antibiotic doesn't fit anymore. We can have some enzymes that come in that degrade the antibiotic before it can ever reach its target. Or we can dislodge an antibiotic that's already bound to the target. So it bound, and before it can work, we want to kick it off. These I don't have a lot of examples for it. It's not a very frequent mechanism. 1 and 2 are the much, much more frequent mechanisms that cells use. Yes, so there's so much research going into that. There's drugs that we can use that can inhibit, the enzymes that they've developed. We can develop new antibiotics that get around these defenses that they've created. We have lots of different ways, and we're finding new ones every day. Just depends on the mechanism. All right, now all of these, they form naturally. So it's not like the cell is sitting there and strategizing and like, okay, well, we've been targeted by this antibiotic in the past, so we need to develop resistance in this way. The cell randomly has a mutation in the transpeptidase enzyme that allows it to be resistant to the beta-lactamase. And that cell just gets the benefit. So this is all due to random genetic mutation. There's no purpose to this. We talk about purpose a lot when we explain this because it's just easier to say it that way. And because we're humans, so when we think about this, yeah, of course I would do this on purpose. But no, all of this is due to random genetic mutations that occur. So really what's happening is if a cell has these random mutations and it's exposed to this antibiotic, it will survive and all of its little siblings will die or cousins will die. And therefore, it's going to be the only one to divide. And now all the bacteria in you have that resistance to this antibiotic. So now you have to use a different antibiotic to kill it. That's the idea here. So let's talk about this. We can keep antibiotics out of the cell. We can use enzymes to destroy them before they enter, things like a beta-lactamase or a carbapenemase. Carbapenems are beta-lactams that are our super, super, super last resort beta-lactam bacteria. So they're like the ultimate penicillins that we only use in very special occasions to save someone's life with something that nothing else is working. Big problem is these carbapenems are so important clinically, and we normally have a lot of strict rules. The whole world has a lot of legal legislation about when carbapenems can be used and how much and how often. One way that carbapenems can never be used is for livestock. And there have been a couple countries where people have gone in undercover and found that they are being given to livestock, and it's causing a big problem with resistance to those now. So our last resort drugs are now being used up and not effective anymore. But both of these, the beta-lactamase and the carbapenemase enzymes, they're enzymes that attack beta-lactam antibiotics and carbapenem antibiotics. They're going to take that beta-lactam ring and cleave it, like we talked about at the beginning. The bacteria says, this is the problem, let's make it not a problem anymore. So these enzymes come in, they destroy the ring, and now they're resistant to that bacteria. So how do we get around this? Because we have to fight back too. So usually we will couple the drug. We'll say we're going to give you a penicillin, a beta-lactam, but we're going to give you something else with it that blocks these beta-lactamase and carbapenemase enzymes. So something like Augmentin, if you've ever had that, it's amoxicillin with something called clavulanic acid. The clavulanic acid binds to the beta-lactamase and inhibits that enzyme. Therefore, the amoxicillin is free to do its work, and it's no longer being broken down. So we have tricks up our sleeve too. It's not just the bacteria. The main way that we keep antibiotics out of the cell, it's a two-part mechanism. We can literally say we're going to block you at the membrane. You can't get past the membrane, or if you do, we're going to spit you back out. So the first thing is we decrease membrane permeability. Permeability just means how things pass through. Gram-negative bacteria, this is more important for them than gram-positives. Gram-negatives have those porins, those channels that they have in their outer membrane that allow things into the periplasm. And one of those is OMP-F. That's studied by Dr. Takahashi here, and she looks at the RNA related to this particular porin. And different mechanisms of manipulating the RNA so the cell cannot get rid of the porins or can't, make more of them, something like that. I don't know exactly what she does. I just know it's related to that. So this is an area that can be exploited by antibiotics that can get in through these porins, and now they're inside the cell. They're like, hey, free opening. Let's take that easy path. So cells, bacteria, what they do is they downregulate the expression of these porins. So if they realize we're being targeted by an antibiotic, they will stop transcription and translation of the porins, and they will prevent more of them from being made, and they'll allow some of them to degrade and fall out of the membrane. So now the membrane has fewer holes in it, it's less permeable. So the antibiotics literally can't even get in. However, this comes at a cost of decreased nutrient uptake. So when you decrease how many porins you have, you're not getting food in either. But the idea is I'll starve for a little while to fight this infection, and when the infection is gone, then I'll start eating again. So it's good for short term, but it's not good for long-term survival. So this is a common mechanism for like Pseudomonas aeruginosa in people particularly with cystic fibrosis. This bacteria is really good at downregulating its porins. So people with cystic fibrosis are more likely to get this infection in their lungs, and then we can't treat it with antibiotics, and they get very sick and can die from it. The second half of this story is we can pump out antibiotics, any antibiotics that do make it through. So up at the outer membrane, if they get in through a porin, we've got these efflux pumps that can pump it back out. So they're called multi-drug resistance, or MDR, efflux pumps. And usually these aren't specific. We don't want them to be specific. We want them to take any kind of crap that gets in and pump it out. So these guys can pump out not only most classes of antibiotics, they can pump out dyes, they can pump out detergents, they can pump out bile, which also disrupts the cell. They can do a lot of cool things like that. So it makes the cell very protected against any kind of toxic chemical, not just antibiotics. One of the main pumps, they're usually made of 3 parts. There's a part that's in the inner membrane, and this is the one that actively grabs onto the chemical and pumps it out. Then there's a protein here that connects the inner part to the outer part. The outer part is usually what we call Toll-C. It's a protein that's found basically in every single bacteria in your gut. And it is always the outer channel membrane of these pumps. And these ones are highly conserved. If there's a way that we can find to target these, we can inactivate all of them across all different kinds of cells, but we haven't really found that yet. We're more working on this part here that recognizes and binds and pumps out the drug. So that's what I was studying in mine. I was looking at all the different pumps with Toll-C and E. coli. It was fun. So these two, efflux and membrane permeability, work together. I'm going to decrease the amount of porins I have. I'm going to decrease the amount of holes and increase the amount of pumps I have, so that way, if anything does get through, I can just get rid of it right away. And that together really makes gram-negatives very dangerous. They're highly resistant to many different types of drugs. They're a pain in the butt to treat. Any questions about that? Cool. There we go. So once they're in, because we can't stop it all, what can we do to inhibit binding? Usually they modify the target. So through random mutations, whatever the target is, like the transpeptidase enzymes, they're going to change slightly. So they can still work, but they can't be bound by the beta-lactam or anything like that. So they can also add modifying groups. They can take like an acetyl group or a methyl group and add it onto a protein and now, or onto the antibiotic. So now the antibiotic can't work. They're like, yeah, we don't like you. You're going to attack us. Let's pop this one little chemical on you, and now you can't function at all. So an example of this is for those aminoglycosides that target the 30S subunit, the cell will acetylate it. So it'll add an acetyl group to it, and then that antibiotic can't work anymore. Oh, there was a question. All right. Cool stuff. And then, all right, any questions about that? That's all the science. We're done with the science. Now we're going to talk about the ethics of it all, which is pretty important, especially if you're going to go into clinical practice. All right. Now, resistance, as I said, has always been a problem. Since the dawn of bacteria, there's been antibiotics and there's been antibiotic resistance. But we are currently making the problem worse in a lot of different ways. We overprescribe and misuse antibiotics like crazy. A lot of people will go into a doctor and have just a virus like a common cold, but they want relief right now. And the last time the doctor gave them the Z-Pak and it worked. I felt better the next day. So give me a Z-Pak now. And the doctors, they don't want to be bullied. They don't want that negative Yelp review. So they just give the Z-Pak. But that's causing now any bacteria that are opportunistic pathogens on you, you're killing the good ones and letting ones that are resistant survive. And now you can then infect someone else with an antibiotic resistant form of a opportunistic pathogen. So there's a big problem. We also, the main culprit is overuse in agriculture. So My scientific grandfather, my PI's PI, he was the one who discovered that for some reason, if you give antibiotics to animals, they get bigger. We don't know why to this day. And not like a little bit bigger, like you get 50% more weight on the chicken or on the cow. And that's a lot more money you can make. And this is not saying, oh, well, the chicken was sick, so I gave it the antibiotics. This is just a healthy chicken. I fed it antibiotics. It got way bigger. So we tend to give a lot of antibiotics to animals who aren't even sick to help them grow more. We also spray antibiotics on plants all the time. We give antibiotics like crazy. I mean, we're doing millions and millions of tons of antibiotics on plants and animals alone in agriculture. So we've got all these soil bacteria that are now becoming resistant to the antibiotics that are constantly being shed by animals or sprayed onto the plants or sprayed into the soil, or the resistance is developing in the animals themselves, and then they infect the humans. Because in particular, cows and chickens, we get a lot of diseases from them. Our immune systems are very similar. So if a cow or a chicken develops a highly resistant antibiotic, bacteria, they can give it to us, we can get sick from it and die, and then we can spread it to all other humans too. A big issue with that is not antibiotic in nature, but viral. We tend to get a lot of really bad viruses from chicken and pigs too. So in general, chickens and pigs are dirty animals and they make us sick. But if we can decrease the use in agriculture, the better. That's a hard one because we need to eat. People need food. They need cheap food. And the best way to do that is to give antibiotics and get more meat out of the same amount of chicken. So legislation is not really a solution here. This is more like a grassroots and a money problem. So if you choose to buy from more small farms that don't use antibiotics, you buy more local, yes, it will be significantly more expensive, but you're going to help combat this issue. You can also make sure that whenever you do go to a doctor, don't take antibiotics unless you have a bacterial infection. Yes, you might feel like crap, but the antibiotics aren't going to do anything to help you anyway if it's viral. So if the doctor says, no, I'm sorry, it's viral, then don't push for an antibiotic, that kind of thing. Yeah. And in the US, we have a lot of strict rules regarding antibiotic prescriptions, and antibiotics can't be sold over the counter. A few of them can, like Neosporin and stuff like that. But for the most part, if you need an antibiotic here, you have to see a doctor. Go down to Mexico, that's not the same case. You can buy them in drugstores. Yeah, a lot of places around the world. It's not a particular country's issue. It's a lot of countries. You can buy Z-Packs just at the grocery store or at a pharmacy without a prescription. And so a lot of people, my mother-in-law being one, whenever she goes down to Mexico, buys a shit ton of Z-Packs and comes back with them so she can take them whenever she thinks that she needs one. That's a problem. So it's an issue of knowledge as well. So if we educate people, that tends to help. The big problem too with that, the flip side of education is you have a lot of places where medicine was used to target people, particularly in Africa. The medical establishment was used to directly harm a lot of Africans. And so there's a huge cultural mistrust of medicine in general in Africa. So even if you do educate and make it where you can only get these prescriptions from a doctor, I'm not going to a doctor. My dad and my mom and my grandmother and my aunt, they were all killed when they went to a doctor. So why am I going to go? So this is not an easy solution. This is a hard problem to fix, but it is literally the number one health problem in the world. It was COVID for a little while, but when we're saying number one problem in the world, we're expecting that in the next 10 years, every year you're going to have 10 million deaths due to antibiotic resistance, up to 50 million deaths every year. So that's a huge problem. We're going to basically go back to the Stone Ages of you get a cut, too bad, guess you'll die kind of thing. So serious problem. I don't mean to scare you guys too much, but it is something that as biologists, we're kind of the the gatekeepers of this knowledge, and we shouldn't be gatekeeping. We need to be teaching people. We need to be the examples, that kind of thing. So there's a thing called antibiotic stewardship, which is learn how to take antibiotics and use them properly, teach others how to do that properly. Know this for the exam. Let me double check that. Sorry. I can't remember if it was this one or something else. Yeah, this is going to be a multiple choice question. All right. And then that is it, I believe. Yeah, that's it for this one. So that was my soapbox. No, I don't